Hello and welcome back to Teach Me in 10. In this episode, we're talking to Dr. Christina Chang, Director of Assay Applications, Spatial Genomics at Takara Bio USA. Christina will introduce a new class of spatial technology that transforms single-cell data into spatial data. As she will explain, the Trekker kit works by tagging each nucleus in its native tissue environment with unique spatial barcodes, allowing each nucleus to be bioinformatically positioned without the use of complex instrumentation. The result is a spatial map with true single-cell resolution, all in a single experiment.
Hi, my name is Christina Chang. I'm the Director of Assay Applications in the Spatial Genomics R&D group at Takara Bio USA. Today, I'm going to tell you about a new class of spatial technology that enables acquisition of spatial and single-cell data simultaneously. In the past 5 to 10 years, there's been an explosion of new spatial techniques that have come out. They can mostly be categorized as capture-based or probe-based methods. The capture-based methods use sequencing as readout. They have streamlined workflows but tend to have lower capture sensitivity. The probe-based methods have high detection sensitivity, but have complex workflows and really expensive instrumentation, which increases the barrier of entry and also limits the sample throughput of the experiments. And for both of these methods, one of the downside is that the data generated is not true single cell, and computational methods have to be deployed to approximate single cell level data. That means if you're working with a subcellular high resolution technique, then you have to perform cell segmentation. But optimal cell segmentation may look different for different platforms, different tissue types, or even cell types. And the noise and artifacts that can be in your data may be hard to detect or filter out, and that can pose challenges in how you can properly interpret data. On the other hand, if you have a lower resolution technique where each spot may be capturing RNA from multiple cells, you have to perform deconvolution. And the accuracy of deconvolution is highly dependent on the quality of the single cell RNA-seq reference, as well as the choice of algorithm. So there's a lot of additional costs and time, both experimentally and computationally, that needs to be invested um, in order to generate a high quality data set. The importance of proper and accurate cell segmentation and deconvolution can't be understated, as many of you are aware. This is an illustration of a T cell surrounded by tumor cells. Currently, you can see that the estimated boundary from cell segmentation incorrectly incorporates some of the edges of the surrounding tumor cells. What this means is that the mRNA from these tumor cells are mistakenly assigned to the T cell, and that might give you data where you could get novel clusters that look like interesting cell types when really they're just artifacts from contamination of RNA from neighboring cells. So what we think the current field lacks is a true single cell resolution spatial technique that can sidestep many of these technical challenges when it comes to deconvolution and cell segmentation. And this is where the Trekker technology comes in. Trekker is based on the SlideTags method that was developed in Fei Chang and Evan McCosco's labs at the Broad Institute. The idea is that instead of fragmenting the information from single cells and trying to reconstruct it, why don't we just preserve the spatial information and then use single cell conventional methods to read out the gene expression information? The fundamental technology behind this method are these spatially barcoded surfaces. They consist of closely packed monolayer of beads, each bead containing a unique spatial barcode, as well as a PCR handle and a handshake sequence that's complementary to the capture sequence on the single cell platform. It also has a photocleavable linker that, upon UV exposure, can enable the release of these barcodes into the tissue. The Trekker workflow is quite straightforward. It takes only 1 hour upstream of any standard single cell workflow. You start with tissue sectioning and placing the sectioned tissue onto the bead array. You then perform UV cleavage to release the spatial barcodes. The spatial barcodes diffuse into the tissue, and then we isolate the nuclei and input them into your single cell platform of choice. That's followed by standard library preparation to generate a gene expression library, as well as a Trekker library that contains the spatial information. After sequencing, the tracker pipeline will use the spatial information to position individual nucleus back to its original location on the tile. The approach takes advantage of the ladder diffusion that occurs during UV cleavage. So the idea is that each cell will receive barcodes from multiple beads, not just one bead, and the abundance of the oligos from each bead should be inversely correlated with distance from that bead. And so you form this distribution of spatial barcodes that can be reconstructed into spatial clusters that the pipeline can use to calculate the weighted centroid. And that's how we determine the positioning of each nucleus. A side benefit of this approach is that you can very easily identify multiplets from your data sets, since they tend to be coming from different parts of the tissue and would occupy and be manifested in multiple spatial clusters. So next, I want to use this example data set to illustrate why we think this approach is unique and has many advantages. This is a example data set we generated from a 25 micron mouse brain section. Right now, we're looking at the UMAP, and this is at this point just a standard single nucleus RNA-seq data set. So you can use existing tools to perform annotation without any deconvolution or segmentation. You can see the UMAP is well separated with all the major and minor cell types identified. But because we have the spatial context, we can also then position these nuclei back to their original location on the tile and reconstruct this very accurate image of a mouse brain. I want to zoom into the astrocyte population in this mouse brain sample. You can see that there are two astrocyte populations with very different spatial distribution. Astrocyte 2 is dispersed across the different cortical layers. And if you have only single cell level data, then you would probably stop your analysis here, because from the UMAP, they would seem like a really homogeneous population. But because we have this spatial context, we can further segment astrocyte 2 based on their location in the different cortical layers, and that led us to identify differentially expressed gene profiles that are unique to astrocyte 2 populations in the different layers. And this can inform and bring about new insights as to their roles and functions in the different cortical layers. This is one example of how having this spatial context can really enrich and bring about additional biological insights from your data. And the type of robust analysis you can perform also relies on the high sensitivity that checker data generates. This is some violin plots comparing checker data to Visium HD data, looking at UMI and genes per nucleus. And you can see that the checker data has at least 10 times the number of UMI and genes per cell. So the sensitivity of the data set, as well as the true single cell nature, allows you to go from data that looks like this, where you rely on deconvolution, and because of imperfect deconvolution and low sensitivity, you have these not well resolved UMAP, to really high quality and high resolution, high complexity data like the ones that I just showed you. Another really exciting aspect of Checker is how easy it is to integrate spatial with multi-omic analysis. This is the workflow slide I showed you earlier, and the only difference is for the single cell assay of choice, we can leverage existing single cell multi-omic assays to also assay additional modalities in addition to gene expression. So we have developed workflows that can integrate Checker with SN ATAC-seq assays, and we have shown that adding the Checker tags does not negatively impact the gene expression data quality or the ATAC-seq data quality. We've also used this workflow to look at more interesting samples, like the primary human melanoma, where we identified a malignant 3 tumor subtype that clusters separate from the rest of the malignant population and also occupies a unique spatial space away from the rest of the tumor subtypes. Differential expression analysis showed that VGF is expressed uniquely in this tumor subtype. And because we also have the ATAC-seq data, we can look at overrepresented DNA motifs found within the malignant 3 tumor subtypes. We also looked at the VGF location, and we can see elevated peak signals coming from this region in malignant 3 compared to the other subtypes, which is consistent with the gene expression that we saw. So with the combination of spatial, gene expression, and attack, there are a lot of really interesting analysis and additional information that you can glean that can inform us of what is happening in the tumor microenvironments. So we think that the tracker technology is a unique solution that can solve many of the current challenges of spatial techniques. From the spatial omics field, this is the first and only technology that achieves true single cell resolution without the need of any computational manipulation of the data. It preserves high sensitivity and gene coverage, giving you high complexity data to get more insight out of your experiments. And it enables very easy integration of spatial with multiomics analysis, which is currently very difficult to do with conventional methods. If you're coming from the single cell side of things, this is a technique that fills in the missing spatial context without changing the experimental and computational infrastructure. And it provides a familiar single cell data format that can make analysis very easy and straightforward. So with that, I'd like to thank you for your attention, and please reach out to the Takara team if you want to learn more. Thank you.
Thank you for joining us for another episode of Teach Me in 10, which was brought to you by Takara Bio USA. Takara Bio offers a wide range of innovative tools designed to accelerate scientific discovery. Their latest Tracker Kit introduces a new class of spatial technology, making it possible to generate spatial and single-cell data from the same experiment without compromising on sensitivity or resolution. For more information, check out the links in the description. And as always, if you liked this episode, be sure to like and subscribe to catch all our latest releases.